I'm going to talk about the most promising cure for diabetes. So let's talk first about diabetes. It's a global epidemic that is affecting more than 450 million people around the world. And so that 1 in every 11 adults actually has diabetes. So diabetes, it's a case of an uncontrolled high blood sugar level. And this is caused by a lack of a hormone called insulin. So this hormone is regulating the sugar uptake by the cells, and this lack of insulin is caused by a very specific case of insulin producing cells death. So these cells are called beta cells, and as you can see here, they are present in our pancreas. So when these cells die in most types of diabetes, then we have a case of diabetes. So, is there a treatment for diabetes? Yes, there are treatments for diabetes, and based on the type of diabetes, but eventually in all diabetic patients, they have to eventually get insulin injections, which is a bit inconvenient for the patients. So how can we actually cure diabetes? And here comes the role of stem cells. So stem cells, from their name, they are the stem from which all the human organs come from. As we can see here, these are the stem cells. And at first, they form the 3 layers of human body. After that, they specify more and develop more to functional organs. So we have here, for example, brains, kidneys, heart, liver, and pancreas. And what we are interested in our lab is that we want to make those pancreatic cells that are lacking in the diabetic patients. So let's see how these cells look like in the lab. So here we have stem cells, and what we are doing is that we are transforming those stem cells into the pancreatic cells that are looking similar to the cells that we have in our own pancreas. And also, as you can see here, the red color is indicating the insulin produced by those beta cells. So now what? How can we test the functionality of those cells? For that, we need to do animal studies, and we implant those cells, those stem-derived cells, into under the kidney capsule of mice. And now these mice are called humanized mice because they are now having cells from a human origin. And also here you can see the implant in white under the kidney capsule of this mouse. And this is a measure of a blood glucose level over time. Can you see it properly? So the red line here, this is a mouse that has not been implanted by our cells. And then when we induce diabetes in this mouse using a chemical called streptozotocin, then this mouse developed diabetes, as indicated here by a very high blood glucose level. However, when we do the same for the mice that have been implanted by our cells, then these mice do not develop diabetes. In other words, curing diabetes. And by that, I would like to acknowledge my lab and also all the funding institutes. And I would like to thank you all for being here, and also the hosts. Thank you.
Thank you, Hazem. Wow. What an inspiring talk, but what I like the most is the stem cell tree. That was pretty amazing. Can you tell me a bit more about stem cells? Can we actually really grow any kinds of organs from them? And where did we get them from?
Well, theoretically, yes. You can grow any organ that you want, but we are still not quite that far yet. So people, like scientists in our research institute in the biomedicum, they are trying to make heart cells, kidney cells, liver cells, blood vessels, everything, but still there is lots of research going on. And Yeah, we are not that far yet. So stem cells, they are coming actually from two different origins. So back in the days, like the first stem cells were coming from embryos. So human embryos that have been donated from in vitro fertilization clinics. So these embryos were donated for research, and then stem cells were derived from these embryos. And then we could use those stem cells because they are self-renewal. So you can use them to get limitless amounts of those stem cells and also limitless amounts of any subtype or subcell that you would like to get. But now we are not doing this kind of stem cell derivation because it's a bit controversial, and lots of regulation actually prohibited that. But now we are doing another technology. So we are deriving stem cells from normal somatic cells. So now we can get any normal cell from your body, like a skin cell or something, and then we can reprogram this and go back into time using a time machine and to bring them back into the stem cell state. And after that, you can bring those stem cells into any kind of cell. And this actually, Shinya Yamanaka developed this technique, and he was awarded the Nobel Prize for this.
Oh, that's good to know. So growing an entire organ is super difficult and slow, and we're not there quite yet, but we can still grow cells, and that is really crucial to your work.
Yes.
Wow. As you said, we used to be deriving stem cells from actual human embryos, and that has something to do with immunity also, because sometimes our bodies can reject new organs that are implanted, or if you get a donated organ, there can be all sorts of issues. How about these grown organs, or even stem cell implants that you grow in the lab? Does our body reject them as well?
Yes. So, like theoretically, if you have an implant from a cell type that is coming from another donor into your cell, your body will reject this cell type because it's coming from another human being. And for the clinical trials that are going through now, that they are giving those cells into so those implants are not rejected because they are giving an immunosuppressant for this, for the recipients of those cells, so that they do not reject those kind of cells. But as I mentioned, those reprogrammed cells. So, for example, if we can take cells from your body and then transform them into stem cells and then make another organ from those stem cells and giving them back to you, then it's unlikely that you will develop an immune response to this organ because it's coming from your own cells.
So it's kind of like your own new organ.
Exactly.
Wow, the human body is amazing. This is also a huge commercial possibility, and a lot of companies are working in this area. Can you tell us something about what the big companies or what the most interesting business endeavors in the field are at the moment? What's happening? So
the most promising company that is working this is called ViaCyte in the US, and they are doing something similar than this. And what they are doing, because safety is the major concern, so they are embedding those cells in something like a device, or you can call it a tea bag. So you can put the cells inside those tea bags, and then you can implant them under the skin of the patients. So the blood can go through those tea bags, and insulin can come out of it in the bloodstream, and by that you can cure diabetes in those patients. And whenever something wrong happens with those cells, you can just take this tea bag out of your cell, of your body.
So diabetes curing tea bags in our very own bodies.
Yes.
That's quite sci-fi, but super interesting. Wow. When do you think, or when do you believe that a treatment based on your field of work will be actually on the market for patients?
Well, we still in our lab, we still have so many questions to answer before going into the clinic. So what we are doing is that we are doing disease models for a type of diabetes called neonatal diabetes, so patients that have been born with diabetes. And what we are doing is that we are trying to investigate the genes that are responsible for this kind of diabetes and try to make models so that we can see what is lacking in the development of those beta cells in those patients.